My name is Albert Wong. I'm a psychiatrist. I treat adult patients at the Center for Addiction and Mental Health just around the corner. And I also work on animal models for psychiatric disorders in my lab. And although the patients I have mostly have schizophrenia, I think the message has already been transmitted that the brain processes and developmental processes are really shared across many different disorders. So I work on animal models. I don't work on teddy bears. I work mostly on mouse. But before I talk about the mice that we work on in the lab, I just want to make the point that the disorders that we see in psychiatry, including things like autism and schizophrenia and ADHD, are among the most heritable disorders out there. So you can see on the screen here, on the y-axis is heritability. And so autism is here, schizophrenia, bipolar disorder, ADHD. And then along the x-axis here is the prevalence. It's the prevalence. And you can see that many of these psychiatric disorders are more heritable than most common medical illnesses. Down here is, so this is type 2 diabetes, for example. This is Crohn's disease, and this is breast cancer. So this is why we're interested in studying genes and the effect of genes on brain development, because they are such a big influence on whether or not people get these disorders. This is another graph that tries to illustrate the kind of genetic structure that many psychiatric disorders, and in fact many common disorders, have. So in this case, the message with this slide is that there are two different main groups of genetic variants that can affect susceptibility to complex disease. On the lower right corner, these are variants that are very common. So that maybe perhaps a third or half of people might carry the variant that's associated with disease. And so these common variants, of course, because they're so common, can't really have such a strong effect on whether or not someone gets the illness. Probably we need many, maybe tens or hundreds of these variants in just the wrong combination to cause someone to have one of these illnesses. Whereas on the top left of this graph, we see variants that are very rare, sometimes so rare that they're only in one family. And these variants, of course, tend to have a much stronger effect on illness. So this is a family in Scotland that had a very large rate of mental illness. So all of the shapes that are colored represent people in this family that have some kind of mental illness. So just to explain how this is laid out, each row represents one generation of the family. And these squares are males and the circles are females. And in this case, you can see there are a number of different disorders. So again, making the point that a single genetic variant can have different effects in different people. And we're not really sure why. This is sort of one big question. So each of the colors represents a different type of disorder. In this case, the most common disorder, and probably the most severe one, was schizophrenia. But there are also people with mood disorder, as you can see, and other diagnoses like substance abuse and learning disabilities. It turns out that this family has a chromosomal translocation, so a fairly severe and obvious, very dramatic genetic mutation in which two arms of chromosome 1 and 11 are switched. And of course, where they switch, they have to first break. And there's a break point on chromosome 1 you can see on the bottom of the slide. I'm not going to use the pointer for fear of disrupting the delicate computer here, but so you can see I'm just going to wave my hand like this. So the breakpoint on chromosome 1 actually cuts through the middle of a gene, just so happens to be almost exactly in the physical midpoint of it. And because this family presented initially with this high rate of schizophrenia, the gene was named disrupted in schizophrenia, or DISC1. So the reason why we're interested in DISC1 and schizophrenia and why we're going to look at the brain, well, I'm going to show you some data about looking at the brain in mice that have mutation in this gene, is because it's been known for a long time that people with schizophrenia have some changes in the brain. And what we're looking on the right is a cartoon cross-section of the cortex. The cortex is the wrinkly outside part of the brain that I'm showing you on the left side of this slide. And it looks like in schizophrenia, which you can see on the very right-hand side of this panel, that the neurons, which are represented by these black squares, they're not distributed in the same way that they would be in an unaffected person. The neurons seem to be squished kind of towards the lower zone, the deeper layers. So the cortex comes in layers. Usually they're numbered in 6 layers. And so in this case, the top is the outside of the brain, the part that we can see, the wrinkly part. And then the bottom is the part that's deep inside. So these layers are stacked together, and they wrap around the outside of the brain. And they're very thin. They're about the thickness of maybe a business card. So imagine a small stack of business cards, 6 thick. That would be the thickness of the cortex around the brain. So I just want to briefly explain to you what we know about how this layered structure develops. So it's kind of a curious pattern. This is the way many things happen in biology. This is not the way that one would build, say, a layered structure in a house. You'd probably start at the bottom and then put something on top of each layer until you have 6 layers. In the case of the brain, it's actually the other way around, meaning that if you look on the left side of the panel, so what's being shown here is a cartoon of the cortex, and each of the columns represents a different time point, with the adult structure on the right-hand side and the embryo starting on the left-hand side. So during the course of embryonic development, what you can see are that these neurons at the bottom are migrating up. You see these little arrows. They wiggle up along a scaffold to get to where they go. And the neurons that, if you see the sort of orangey colored neurons that are being born fairly early, you can see that they end up in the deep layers near the bottom, layer 5 in this case. Whereas there are some yellow colored neurons which are born nearer to the right-hand side of the panel, so much later in development, and those neurons end up in the top layers. As you can see, they're in layer 2. So this is a kind of inside-out lamination pattern. And this is relevant because the neurons that are born late have a very short time to migrate the greatest distance. So they have a very challenging task compared to the neurons that are born early, who have, they can proceed at a much more leisurely pace, and they only have a short distance to go. So we think that if there are problems with genes that code for proteins that affect the way in which neurons migrate, or the machinery, the motor that drives them to migrate, that maybe this could affect the way in which the cortex forms. And so this is what we wanted to test. And specifically, we were interested in this because the DISC1 protein seems to interact with a number of other parts of the cellular machinery that drives neuron migration. So the fried eggs that you see on the bottom left of this figure are meant to be embryonic neurons climbing along the scaffold to get to where they go. And they kind of go in a stepwise fashion, where they stick out an arm, it attaches to the scaffold, and then there's a bunch of fibers inside that retract, that pull the yolk, or the nucleus in this case, towards the leading process. And then it detaches, and then it goes again. So step by step, up through the radial scaffold towards the target. So DISC1 is a protein that's part of those colored shapes that are around the nucleus there, around the centrosome. And it seems to regulate the way in which these fibers, they're called microtubules, how these fibers contract, and therefore the engine that's pulling the nucleus and the cell towards its destination. So what we thought is that it's possible that mutations in this DISC1 gene, because it's related to many kinds of mental illness, and because we know that schizophrenia has changes in the structure of the cortex, perhaps mutations in the DISC1 gene might also affect both the structure of the cortex and its development. So these are 2 students in the lab, Frankie and Mark, who did an experiment with a mutant mouse that has a mutation in the DISC1 gene. I think it's not totally clear. I'm just going to try the lights. So what you can see, so there's 2 rows of figures. The top one shows a cross-section of the cortex. And then there's, on the left is the wild type, the sort of normal mouse without the mutation. And then on the right panel, you can see some of the mice that have mutations. And what you can see is that the mouse on the left, this is a cross-section of the cortex. And we've looked at cells that are supposed to migrate to a superficial layer. And you can see that on the left side, it's a very tight band, and that's the way it's supposed to be. But on the right side, you can see that it's kind of more of a sprinkling, indicating that the neurons didn't get to where they were supposed to go. We also looked at another type of neuron, which you can see in the bottom panels. On the bottom right, you can see a cartoon of where the neurons are supposed to go. They're supposed to sort of sweep across in an arc from the bottom right to the top left. And these are called interneurons, they're a different kind of neuron, but the principle is the same, that they also have to sort of crawl through the developing embryo to get to where they're supposed to go. And again, you can see that on the left panel, the wild-type mouse, you can see that the neurons, the green labeled dots, they have gone a lot farther towards where they're supposed to go than in the middle panel. So again, both of these kinds of experiments indicate to us that the mutation in DISC1 does affect the way that neurons migrate. So that's consistent with our idea that one of the reasons why DISC1 might be causing mental illness is that it's changing the way, it's impairing the way that neurons migrate and develop. So another big question in medicine in general, in terms of complex disease, and specifically in psychiatry, and relevant to all the disorders that have already been mentioned today, is this question of resilience. So this is a concept this is not my concept, this is actually, I think, a Swedish concept that Dr. Boyce, who's a pediatrician at UBC, likes to talk about, I think sort of popularized this idea of dandelions and orchids. So we all know children, perhaps some of us are lucky enough to have children like this who are very resilient. So they're kind of like dandelions. They can grow anywhere, they don't require any special care, they're very tough, and left to their own devices, they are just fine. And then, of course, we also know children who are like orchids. They're very delicate, and they require just the right conditions. There's another scientist at U of T, Marla Sokolowski. She's also a geneticist, and she likes to describe this orchid phenotype in one of her own children. And she says that this is the child who, the whole day can be ruined by itchy socks. So, things have to be just right for these kind of children. And, of course, we know adults like this too, where the slightest perturbation can derail the day. And so the message here is that the same kind of stressor, whether it's something silly like itchy socks, or whether something more serious like a mutation in a gene, doesn't have the same effect in everybody. Whether it's the genetic background or interactions with the environment that might mediate this was not really clear. So this is just a lead up to some experiments that other students in the lab, Tatiana Lepine is actually a postdoc in John Roeder's lab, who was the one who developed this mouse mutant model in the first place, and Nipa is another student. And they did some experiments looking at the interaction between the mutation in these mice and environmental factors that we know are related to mental illness in humans. So in the top you can see something called social defeat. This essentially represents the kind of, it's a model in animals to generate the kinds of events that are stressful to people that tend to cause depression, or at least precede an episode of depression. And in the bottom panel, you can see an example of an experiment using something to simulate the effect of infection during pregnancy. So we know that certain kinds of infections during pregnancy can also increase the risk for brain disorders, including autism and intellectual disability and schizophrenia, for example. And so what Tatiana and Nipa found was something like this. And so I know you can't read the text here very clearly, but the message really is that each of these lines in each of these panels represents the effect of environment on a particular genotype. So each line represents a mouse of a different genotype. Sometimes it's a mutant mouse, or sometimes the normal wild-type mouse. And each panel represents a different kind of behavioral test. So it's not so important what these specific tests are. What's important is that you can see that the relationship of the lines in each of these panels is different. Sometimes they're parallel and sometimes they cross. And when they cross, it means that the environment has a different effect on that behavioral readout, depending on genotype. So there's a gene by environment interaction. So, we didn't really figure out exactly how this occurs and why it occurs only in some behaviors and not in others. But I think it does capture the idea that different types of behaviors, or in humans when we call them symptoms, are affected differently by environment and gene interactions. So finally, I want to tell you about how we've used this mouse model to test new treatments. And I think this is, this is very early kind of data. It's not something that's ready for testing in patients, but I think I'm trying to give an example of how mouse models can be useful in developing new treatments, and to hopefully give you some hope that there will be advances, hopefully not too long from now. So again, Tatiana and Nipa did was to, sorry, I can't go back, but what they did was to treat some of our mutant mice with a drug that's already used to treat seizures in people. It's called valproic acid. And so one thing I didn't tell you about these mice is that they have a trajectory, they gradually develop abnormality. So they're fully abnormal around the equivalent of late adolescence in humans. But before that, in early adolescence and around puberty, they're not really that abnormal. So there's sort of a certain time that seems to be critical to allow these abnormalities to kick in. And so what we wanted to do is give a medication before this happened to see if we could interrupt that process and maybe repair the process of development. And that's what we saw. I won't go too much into these data, but basically the message is that many of the abnormal behaviors that we saw in these mice is prevented even after the drug is stopped by the treatment with valproate. And also we found some changes in the brain. That's what the panel on the top shows. We saw some extra glial cells. These are sort of the supporting cells in the brain that are sometimes activated during inflammation. And we saw that in the mutant there are more of these cells than normal, and that the valproic acid was able to suppress them. So this treatment, this sort of pre-treatment, if you will, this kind of preventative intervention was able to change the sort of path of development so that these mice were more normal than they would have been without the drug. And so what I want to, the reason why I'm going into that example is because I want to sort of introduce the idea that it's possible to perhaps repair or fix structural changes in development even after the developmental event, the critical period for that developmental period has ended. So these are not our data, this is from another lab, but it, I think, illustrates the point. This is another mouse mutant that has a mutation in a gene that also affects cortical lamination called DCX, or double cortex. And what you can see in the middle panel is on the top of the middle panel, those blue dots are supposed to be the normal neurons in the correct location in the cortex, near the top. Just like that picture that I showed you with Frankie showing the green dots near the top. The middle panel with the red dots shows what happens in the mutation, that these same neurons are seemingly not able to get to where they're supposed to go, and they're stuck in the deep layers. And so the question is whether or not re-expressing this gene is able to make them go back to where they're supposed to go. That's what the question mark is supposed to show. And so the results are that it is possible to sort of repair this problem later on. So depending on when the gene is re-expressed, so basically they have this model where they can switch the gene on and off. Ours is just off all the time, or it's just mutated all the time. But this one, they can turn it on and off. And basically they show that if they turn the gene on even after birth, even though the cortex is laminated during, in utero, while the fetus is still in mum, even though you turn the gene on after the baby is born, in this case the mouse baby is born, you can still get some partial correction of the phenotype. In other words, the neurons can still be induced to migrate back to their proper location. Not perfectly and not all the way, but at least part of the way. Some of the neurons go part of the way. So I think this is very hopeful, the idea that even though there may be some abnormalities in brain structure that occur very early in life, before anybody has any idea that there's anything wrong, it's not necessarily fixed. It doesn't mean that there can't be some attempt to induce these neurons to, again, say, migrate back to where they're supposed to go, even though it's way past the correct time that these neurons are supposed to do this. So this is the last slide. I just want to show you that represents another approach, again using animal models to try and illustrate how this can be useful, how these models can be useful. So this is another student, Joel, who's testing new drugs. And he's testing drugs from a consortium called SGC, which stands for the Structural Genomics Consortium. And this is a collection of academic and industry partners that is trying to figure out the structure of various proteins that are involved in regulating DNA, and also trying to find small molecules or drugs that can change or inhibit the function of these proteins. And so the main focus of the SGC has been to look for new cancer drugs, because the molecules, the enzymes that regulate DNA are very important in cell division, and they represent a good target for killing cancer cells. But you may be aware that psychiatry has a long history of repurposing drugs from other areas of medicine. So iproniazid was an anti-tuberculous drug, and that was the first chemical that was noted to have antidepressant effects. And so the first antidepressants were based on this observation that an anti-infection drug, that an antibiotic essentially, was useful for treating some kind of behavioral change. Chlorpromazine is the first antipsychotic. It was repurposed from a drug that was originally designed to augment anesthesia. So we're following in that tradition by looking at drugs that are targeted at cancer, but seeing whether or not they might be useful for treating behavior, because there's also evidence emerging that these proteins that affect DNA, that regulate DNA, also may have an effect on behavior. And so the graph there is kind of complicated, but the message there is that we've found that some of these drugs that were designed to kill cancer cells also have an effect on behaviors that are related to psychiatric illness, the same kinds of behaviors that we saw that were abnormal in the mutant mouse I showed you earlier. So, there's no, obviously, solution to clinical problems immediately, but I think, I'm hoping that I've shown you a few areas in which research using animal models can help advance the field and perhaps eventually lead to new treatments. Thank you.